Hi guys, welcome to the channel. So in the previous video we were discussing about the elemental analysis and we discussed the classical procedure for elemental analysis. So in that procedure we discussed that we have to perform three types of analysis, that is quantitative analysis, qualitative analysis and molecular mass determination. So qualitative analysis and quantitative analysis, if we combine the information of these two, we can get the empirical formula of the compound, and empirical formula combined with the molecular mass, these two informations combining, they can give us the molecular mass of the unknown compound, and then after we can proceed for finding the molecular structure of the compound. So these were the three main steps of the classical procedure, that is quantitative analysis, qualitative analysis and molecular mass determination. Now one by one we will discuss each of these. So first we will move on to the qualitative analysis. In qualitative analysis, as we have discussed, that we are going to find out which types of atoms are present in that unknown molecule. So if we have an organic compound, then we are going to pre-assume that carbon and hydrogen are going to be already present in them. If you want to show that carbon and hydrogen are present, just simply burn that compound in the presence of excess of oxygen. So what happens if we are burning an organic compound in the presence of excess of oxygen, then carbon dioxide and water molecules will be released. If carbon dioxide is released, that means carbon is present. If water molecules are released, that means hydrogen is present. So if burning of organic compounds results into the formation of carbon dioxide and water along with any other product, then that means carbon and hydrogen both are present. Now the next question is how we are going to detect the CO2 and H2O after the combustion process. So for that we have gas chromatographic technique. So if the original sample amount is known, then also we can determine the relative amounts of carbon and hydrogen present in the organic compound. So GC is a very sophisticated type of technique. So using that we can easily determine the amount of carbon dioxide and water released, and if we know the amount of original sample, we can also determine the amount of carbon and hydrogen. Now this is all about qualitative analysis, in which we can find out the type of atoms present in the molecule. After that we move on to the quantitative analysis. In quantitative analysis we will try to find out the amount of each of these atoms present in the unknown molecule. So for that what we are going to do is we are going to take the help of the instruments, the modern instruments. So these types of calculations in which we want to find out the quantity of atoms in the molecule, these type of calculations are already performed by the instrument itself, but our main part is to understand the basic principle behind these type of calculations. That is our main part. So we should know what are the principles involved in the calculation of the amount of each of these atoms which are present in an unknown molecule or organic compound. So I'm going to link an example. In that example, if you're going through all of that example, you're going to understand how we are going to analyze the amount of atoms when we have certain type of data given which is provided by the instrument like GC or any other instrument which is going to provide us some data, and using that data how we are going to calculate the amount of each of these atoms present in a molecule. So have a look at this example, try to go through all of it and try to understand that. You will be able to understand that very easily. So I would highly advise you to go through that example, try to understand that example, that how the calculation, the system of calculation in the instruments work. So we are done with the qualitative analysis and quantitative analysis. Now moving on to the molecular mass determination. Now molecular mass is determined nowadays by using mass spectrophotometry, or you can say mass spectrometry, not photometry, spectrometry. actually we are going to study about mass spectroscopy or spectrometry in a separate chapter. There is going to be a separate chapter about mass spectra, so we will discuss in detail in another video of mass spectra. So these three procedures are done: quantitative analysis, qualitative analysis, and molecular mass determination. So now we are left with very few of the topics in this chapter. With that basic foundation before starting out the spectroscopy will be over. From next video onwards, we are going to discuss some of the classical methods to find out the molecular mass. So initially, when mass spectrometry was not developed, we were having some of the methods to find out the molecular mass of the unknown compound. We are going to discuss that in a very short way, and then we will move on to the next topics. After that, we will try to find out the molecular formula of the compound. Then we will discuss what is index of hydrogen deficiency, and then we are left with one or two small topics, and then this chapter will be over. So I hope till now everything is clear to you guys. So far that's it for this video. Thank you so much for watching.